All right, well first I want to say thank you for everybody for coming today. The purpose of this is these young people are going to be presenting at Argonne National Labs to the scientific community the findings that they did for their research project. And when they're there, scientists, other students, faculty from other places are going to come up and they're going to ask them a lot of probative questions about, you know, what did they do, how much they understood. So that's why you all have a piece of paper in case you want to ask them questions. It's going to help them sound like the intelligent people that they are. I just don't want them to be surprised by questions, and I want them to, you know, have some ability to think about them before they're on the official stage. This one, I count this as an official stage, but that one's the real one. So let's see, we'll start with Max. All right, so Max, this is Max Bacon. He's a senior, and he has been on the ESRP team for 3 years. Next year, he plans on studying computer science at the University of Illinois at Urbana-Champaign. He plans on becoming a software engineer to apply programming skills to complex problems. Max was a recipient of the Air Force Junior ROTC J100 Scholarship, which is an ROTC scholarship providing a full ride scholarship to any school for 100 junior ROTC cadets in the nation. He enjoys playing guitar and baseball in his free time, and he looks forward to traveling and spending quality time with friends this summer. I should mention, Max joined our team as a sophomore. He came in every morning and got tutored on chemistry before he was anywhere in chemistry. So he learned chemistry ahead of his chemistry class just so he could participate in this program. Thanks, Max. Next we have Eliza. Eliza, stand up please. Eliza DeAnching is a senior from Lincoln Way East. Eliza is involved in extracurriculars like varsity tennis and badminton. She's part of Science Club, International Club, Future Health Professionals Club. For awards, Eliza is part of NHS. She's an AP scholar with distinction. She is she has the Spanish biliteracy seal and varsity tennis sectional champion. Eliza is committing to UIC to pursue pharmacy and to become a clinical research pharmacist, where she will direct and develop research on existing and new prescription drugs. This summer, Eliza is going to LA for a month of fun before college. Also, she was a champion in SWSC science competition. She forgot to put that on her bio. Divya? All right, so Divya, I'm gonna have you at Divya. Divya is a senior, and this is her second year in the ESRP program. She is involved in several other extracurriculars at Lincoln Way East, such as mathletes, chess team, and varsity badminton. In terms of awards, Divya received the National Merit Committed, the Biliteracy Seal for Spanish, and an AP Scholar with distinction. Currently, Divya is committed to UIUC for computer science and bioengineering, aiming to learn more about the interaction with technology and the biological field. Over the summer, Divya is excited to be going on a trip to India for almost a month. Thanks. She also forgot to write down she's a champion for the Southwest Suburban Science Competition, but that's all right, Divya. Sophia Giampaolo will be attending St. Mary's and Notre Dame for their 5-year dual degree program, in which she will receive a degree in chemistry from St. Mary's and chemical engineering degree from Notre Dame. Sophia was awarded the Presidential Scholarship from St. Mary's for her academic achievement. During high school, Sophia has been active as a captain of the Lincoln Way East Scholastic Bowl team, ending her 4-year career as one of the top 10 players in school history in the fine arts category. She also participates in Creative Writing Club, the Frankfurt Library's Teen Advisory Board, and of course, Science Club. When she is not in the laboratory, she also enjoys drawing and fashion design. Sophia has won the Illinois State University's annual fashion design competition in the high school category. She hopes to spend this summer knitting, playing with her cat Taco, and working at her part-time job at Aurelio's Pizza. Sophia would like to thank her family, friends, and God for supporting her passions. Next we have Madeline. Madeline Dreyer plans on attending Johns Hopkins University and majoring in cellular and molecular biology or biomolecular engineering. She hopes to pursue either cancer research or immunology studies. Madeline is the president of National Honor Society. She's an associate to the Girl Scout Board of Directors and has leadership in bringing change to mind and future health professions. This summer, Madeline looks forward to going to the Bahamas. We have Griffin next. This is Griffin Reedy. He's a junior, and this is his first year working on the ESRP team. He is my only junior, so we're going to be refreshing our team a little bit next year with some younger blood. He's going to be probably a leader for this program next year, provided our participation is never guaranteed. Every year we have to reapply to this program, so we're still hoping that we will get another project for next year. This past year, he has participated in Science Club, Griffin Crew, Peer Helpers, and has held a part-time job. He has a goal of attending University of Notre Dame and majoring in electrical engineering. This will hopefully lead him to a career in technology with focus on quantum computing. Currently, he is waiting to hear back from the admissions committee with Notre Dame Leadership Seminar program for this summer. Additionally, he will be going to Uganda for a week and a half in June to build a park and distribute food in a refugee camp. Thanks, Griffin. Next, we have Alex Korzowski. Alex is a senior, and this is his first year on the ESRP team. He will be studying computer science and statistics at the University of Illinois Urbana-Champaign next year with the goal of becoming a software engineer. Alex is also a member of the autonomous car team, which we took first place in both divisions last year with his participation. He also is involved in the Samsung Solve for Tomorrow. With his idea, we were state finalists, and we won some products or some prizes from Samsung for his efforts. Outside of science, he's a member of Art Club, Mathletes, and various other activities. This summer, he and his family will be taking a 1-month vacation to Japan and Taiwan. Thanks, Alex. Last but not least, we have Drew Clarkin. As a student at Lincoln Way East, he has participated in varsity volleyball, science club, coding club, and robotics club. Next year, he will be attending IU at Bloomington with a merit scholarship and plans to major in both computer science and bioinformatics. After college, he plans to work in the bioinformatics industry and focus on fighting the planet's environmental problems. This summer, he plans to complete a personal project with a focus on climate crisis software to propel his career goals. Also, he is an Eagle Scout, which I am too, and so I really like that about him. So, in order for kids to become part of this program, they go through a pretty hard interview process, and it's super hard for us to go through this process this year. Maria, our department chair, and Nicole Cashin helped me select the students, and Nicole has been my partner in this whole project. So Nicole, either stand up, wave, whatever you want to do. All right, well, without further ado, I'm going to let them take over.
So we were part of 2024 Argonne ESRP team. Is this on? It's on.
Is the switch turned on the side?
Yeah, it's on the side.
On here.
It says it's reading, eh? Check. Oh, you know what? Sometimes it. Wait. Now we'll go back. Now try
Oh,
it.
there you go.
Oh, there you go.
Okay.
Alrighty, and here's a picture of the team. Our mentors, Mrs. Forrester and Mr. Podowski, were our mentors for this research team. All of our team is here except for Liette Zacharias. She's absent today. And our mentors for the school were Mr. Volva and Mrs. Cash. Our motivation came from our mentors' research paper that they worked together with other Argonne scientists, which was called the NA22. In this research paper, they investigated the possibilities of tracking radioactive chemicals in soil over a past period of time. And this would allow them to see in foreign countries whether or not there were radioactive materials in an environment, such as weapons, for the government to find. In their analysis, they found that certain Purex chemicals, plutonium-uranium reduction extraction chemicals, were found to do considerable damage to the soil microbial community composition. And they concluded that there was a possibility to use the soil and the chemicals that they found to identify whether or not there had been radioactive substances in the area in a past amount of time.
So moving on from that, sadly we can't actually work with radioactive substances because we're a high school team. So instead what we did was we worked more with household pesticides. And what we did was we want to measure a similar trend of seeing how these pesticides affect soil, but a more scaled down high school level where we can really focus on the process and learn from it.
So with our mentors research project in mind, we sought out to create a project that looked at different insecticides, pesticides, herbicides, and fertilizers and see how they affect concentrations of different amplicon sequencing variants within microbial communities. And the first step of that process was identifying 5 of the biggest chemicals in the agricultural industry as of now.
So one of those chemicals that Griffin was talking about was gamma. It's a man-made organophosphate and also a restricted use pesticide, which means that you must have a license to use it on a farm. It's an acetylcholinesterase inhibitor, which means that it prevents the breakdown of acetylcholine, which creates an excess amount of acetylcholine in the soil. It's commonly used with soil-dwelling insects on farms, such as nematodes and wireworms, and it's applied to the fields using either the irrigation system or granules. And while it is helpful for getting rid of pests, it is also very harmful towards humans, animals, and the environment, causing effects such as nausea, vomiting, dizziness, difficulty of breathing, and even possibly death.
Moving on from that, we have another chemical, this one being malathion, and this one, like gamma, is a man-made organophosphate, and similarly it is an acetylcholinesterase inhibitor. But the primary difference between this one is the targeted insects, with gamma being focused on ground-dwelling insects, and this one being fruit flies, mosquitoes, and even some lice with shampoo applications. It has similar negative side effects with including suffocation, paralysis, and eventually even death of insects.
Next we have atrazine, and this is a fine powdered herbicide, and it's often mixed with a carrier to be applied to fields. Often it's used with cornfields. Again, it's classified as a restricted use pesticide, and it works to block chloroplast electron flow, thus limiting photosynthesis and killing the plant. Although there hasn't been a lot of research done about how it affects humans, there has been research done among animals where it shows that reproductive issues can occur after long-term exposure.
One of our other chemicals was fipronil, and it is an insecticide that disrupts an insect's central nervous system. It is a slow-acting poison, so when an insect goes to their colony and they die there, it can also kill other insects in that colony. And it is a pyrosyl class of molecules and is a amino compound, sulfoxide and benzene. There's not much research that has been done to show if it has any effects on humans.
So another one of our chemicals was urea, and as you can see here, its molecular compound is CONH2. And so you can see right here in the model, we have 2 amino groups and 1 carbonyl group, which is just the oxygen double bonded to the central copper atom. And it can be considered an amide because it has a carbonyl group bonded to a nitrogen atom. Urea is also functionally related to a carbonic acid, which means that when carbon dioxide dissolves in water, it can be a weak acid in like very small amounts. And it has very widespread applications but it serves as a human metabolite. It's a starting material in many plastics and drugs. It can be used as a feed supplement, and most importantly to our project, it is one of the most concentrated nitrogenous fertilizers. And it's so nitrogenous because it's like the chief product of of the metabolic breakdown of proteins in most mammals and fish. And as you can see in like its compound, it's synthesized from ammonia and carbon dioxide in the liver, and it's secreted through the kidney, and because urine is like mostly made up of urea. And something else important about urea that will be important later is that through hydrolysis or nitrogen fixation, it can also become very acidic to the soil microbial life. So, but we'll come back to that later.
So we're going to begin with our sample collection, and for that we were given 100 samples that we could create, and we used soil from Fermilab, which is near Chicago, for the soil. From there we took the soil and picked out any sticks, any seeds, anything that wasn't dirt. It was kind of satisfying. And from there, the chemicals, we then made 10 molar solutions of all 5 of the chemicals. Then from there, diluted them into 1 molar and 0.1 molar solutions that we then added to our samples. And as far as controls go, as I said before, all of the soil was from the same location, Fermilab. In addition, all of the soil masses in each sample were the same, and all of the solutions we added were of the same volume. And with 5 chemicals tested times 3 molarities times 6 samples for each, with our 10 negative controls, we had a total of 100 samples. And then after that, we were able to start extracting the DNA from our soil. We used the DNeasy Power Soil Kit, which is very nice that we are able to have this available, since it has been in the past hard to collect DNA from soil. The soils were then they were placed in a test tube, and we had in there some beads and a solution. And when you took that solution and everything in there and put it in a centrifuge, that helped break up the soil, and it actually the beads in there were able to remove the organelles and nucleus and all the DNA that we needed out of the cell. So then from there, we were able to take out the layer that had from the centrifuge that had the DNA components, put it in another container, add some additional chemicals, put it in the centrifuge, and then you keep repeating that almost like a filter until we had a few drops of clear liquid which had our DNA inside of there. And then we can start analyzing it and start our conclusions.
So once we've completed choosing our 5 chemicals, as we stated before, and the sample collection, and then the data extraction, we move on to sequencing, because now we have to figure out what bacteria are actually involved in the soil and the microbial community. And so this led us to using the 16S sequencer, and this was a vital piece of equipment that we had to rely on this year. Last year that was the advanced photon source, but this year, since we are trying to sequence DNA, we would be relying on the 16S sequencer. So what exactly is the 16S sequencer? It is a form of amplicon sequencing, meaning that it focuses on a targeted region to zone in on a specific genomic region and look for genetic variation within that specific area. And this sequencer, as it has in its name, focuses on the 16S rRNA gene, and this is present in all bacteria and archaea. And what makes the 16S rRNA the preferable gene that we focus on is that it's the 16S subunit of the ribosome within a particular specimen. And the reason why it's the one that's mostly used for microbial classification is that it can identify variation within the genetic level at 0.5 to 1% divergence, you can see over there. And essentially it has 8 highly conserved regions, which have a high level of taxonomy, meaning they have a lot of bacteria involved in them, a wide variety of it. And then they also have 9 variable regions. And because of this divergence, it allows you to differentiate between the numerous amount of species of bacteria and archaea available, and just makes it overall easier to identify those differences. And so how the sequencer itself actually works is that it amplifies the 16S rRNA sequence, or that gene within the DNA, using a process called PCR, which is known as the polymerase chain reaction. And you can see that kind of happening in this video where, we can come back to it when it refreshes, but the PCR is performed by using primers on a particular region known as a hypervariable region, and as the name indicates, it has a high variability in the bacteria in that particular region. So here you could see that like it selected using that primer the part of the gene that we want to focus on, and then it chooses that specific region. So the green and the yellow are the focus of where it's going to start comparing those base pairs. This way it's more efficient so that we don't have to go through the 1,500 base pairs that are actually involved on average in the 16S rRNA gene. But essentially it compares between those base pairs, and then if it accepts those, it sees that it's correlated to a particular type of bacteria, and then we can identify that, as Griffin said at the beginning, the amplicon sequencing variant, which is an ASV, that's like the placeholder for the true bacteria name. But it allows you to identify what that ASV really is. And another interesting fact about sequencer is that you can change what region of the 16S rRNA you want to focus on based on what type of bacteria you're looking at. So moving on, one of the big problems that we faced with our project was that sometimes we didn't know exactly what specific bacteria it is that we're working with, because there were thousands of these ASVs, like we mentioned before. So because there's thousands of these bacteria involved in the microbial community, we needed to identify what specific strain we're working with. And so when we used an application called RStudio, and we'll talk about more of that later because it was a big part of our data analysis. But RStudio allowed us to create a full table. After we imported the data from the 16S sequencer, we were able to import that data into RStudio and create a full table of all the different bacteria involved within the microbial communities of the samples that we had. And so using that table, it was able to classify each of those bacteria into their phylum, class, family, and genus. And so of all of those, sometimes it wouldn't be able to identify the specific, it wasn't able to classify to its highest level. So sometimes it would just, phylum being the lowest, it would just identify phylum or class, and it wouldn't have the order or genus, which are a higher classification. So what we had to do was we had to sometimes take the DNA sequence that we imported using the data from the sequencer, and we had to compare that on a website known as BLAST. And what BLAST is it's a database of all the different 16S rRNA DNA sequences of all the bacteria and archaea in the world. And so that database essentially compares whatever DNA sequence we took from RStudio, and it compares it to each of the sequences that exist in the world, and it allows us to identify what bacteria is closest to that ASV. So this is the DNA sequence that we got using RStudio, and we essentially used this, put it into BLAST, and then this first line here, I believe, is our actual DNA sequence that we inputted, and then it compares it to possible base pairs of other bacteria from their sequences, and in this case, it matched up best with Pseudomonas of this strain, and so we know that now this ASV particularly identifies with this bacterial strain, because it was a 100% match when we compared it. And to have a better visual, so this is like the like a chunk of DNA of this specific strain over here, and you can see right up here, this highlighted part matches to this DNA sequence, which is what BLAST is essentially doing. It's taking this DNA sequence, matches it up with it to identify what it is. And another useful aspect of this is that sometimes in RStudio, when it creates that table, for one of the ASVs, it would identify it up to the genus, but then for another one, it would have the same type of phylum and class, but then it wouldn't explain what order and genus it has. So then the problem with that is we don't know if they're the same thing, so we don't want redundant data. So what we can do is take that same DNA for the other bacteria, put it in here, and see if it matches to the same strain. That way we're not using redundant data over and over. And then now we move on to part 4 of our project, which would be the data analysis process. And so this is where RStudio was especially helpful, along with the other aspects of it. So what we did with RStudio is we created box plots using the data that we imported, so that we could see how, based on the days and concentrations, the concentration of each bacteria, the ASV, is altered. So if you have to turn it on.
Did it stop working? Yeah, touch the screen.
I think I might have to. Is it okay if I just talk from here for this one?
Sure.
Or- okay. So for this part right here, so this is the code. I know it's a little bit hard to see, but the first 2 lines are really what's important. So the first line, the x equals chemical, shows what we're going to be focusing on, which would be the so the x-axis would have the 5 different chemicals, including the control group, and then the y-axis would have the actual ASV that we're focusing on and the concentrations of it. And then right here where it says date is showing how we're going to change the way we're going to organize the box plots. So for a visual. So this is the box plot arranged based on days. As you can see, day 0, day 35, and day 7. And so on the y-axis here, we have the actual ASVs, so we could change which bacteria we're focusing on, and this is the concentration of it. And down here on the x-axis, we have the actual chemicals. And so these box plots essentially show how that concentration varies from day 0, 7, and 35. And the lines here, you would just read it like a normal box plot. It shows the range, some outliers for the actual concentrations, and the middle line here shows the median for concentration for that box plot. And so this was vital for our project because we were able to use this to get a easy visual to see how the patterns in the bacteria were changing based on how long a specific chemical was in the soil, or in another type of bar graph, which is organized based on concentration. So this one is based on concentration. We were able to see the patterns, like in this case we see Malthion and urea have a significant decrease in a higher concentration levels, and we will reference those in our findings. But this graph really helps us see how those patterns emerge as we look at them.
Alrighty, so in our findings for gamma, so gamma is right here. As you can see, day 7 is in the blue and day 35 is in the green. So the concentration from day 7 to day 35 greatly decreased, and it seems like day 35 went back to around the same concentration of the control substance. And one of our reasonings for this would be microbial degradation of the soil, which would lead to a decrease in the concentration of gamma in the soil. And the reason that could happen would be due to the property of gamma being an acetylcholinesterase inhibitor, which would prevent the production of ACH, acetylcholine, and this would lead to an increased rate of soil decomposition and would negatively affect the soil health by decreasing the nutrient cycle and soil fertility.
Continuing on for more findings for gamma, something we noticed here is you'll see that, I think it's point working, there we go. So for gamma, we'll see that day 7 goes up to day 35, but atrazine and fipronil follow similar trends where they'll both increase while Malthion and urea are different. And the same goes for ASV14 here, where we'll see similar trends between gamma, fipronil, and atrazine. And what we concluded here is that it's likely that the mode of action where gamma inhibits atrazine, acetylcholinesterase, atrazine disrupts photosynthesis, and fipronil interferes with the nervous system. These modes of action were very likely affected similar parts of this bacteria and resulted in similar trends for these 3. And although this wasn't like a universal trend for everything, it was very common for us to see atrazine, fipronil, and gamma following very similar relationships.
Next we have Malthion for our findings, and so like I said before, Malthion for concentration shows significant decrease, which we'll reference in the next slide. But for days, it seemed to be that at day 0 and day 7, there always seems to be a significant increase or decrease based on what bacteria we're focused on. But then after day 37, where it just the duration increases, it seems that Malthion always seemed to return to its control levels. As you can see right here, this matches kind of with like the control levels over there. And so because of this dip, when we looked into like Malthion's properties and stuff, we found that it has a high degradation rate, meaning that it has almost like a week in which it lasts in the soil before it breaks down completely, and its effects are rendered are as effective anymore. And so because of this high degradation rate, Malthion seems to return to its original, change the concentration levels of that bacteria back to its original after that first week of initial increase or decrease.
And just as Divya referenced earlier, the concentration tends to be a more significant factor in terms of how it affects bacteria. With higher concentration of Malthion, specifically 10 molar, we see that we have a dramatic decrease in the concentration of bacteria, specifically ASV6. And this is due to, with higher concentrations of Malthion, we get lower production of CO2. And because CO2 is very crucial for the bacterial community within the soil, it allows plants to thrive. And when deprived of CO2 and the application of Malthion, we see significant decrease in ASV6.
So our findings for atrazine were that from day 7 to day 35, there's a decrease in ASV6, which using the blast nucleotide we found was to be hyphomicrobium. And hyphomicrobium plays a big role in initiating the DNA replication process. And we know that since atrazine is effective in disrupting electron flow, which is essential for DNA replication, atrazine is basically inhibiting the main function of the ASV6, which is hyphomicrobium, and that is the main cause of, or a main cause of the decrease. And then our continued findings of atrazine are that at the 1x concentration of atrazine for ASV31, which is alpha protobacteria, there's a large variation in the concentration between samples. And this could be due to the fact that alpha protobacteria is found like in the deep ocean or in glaciers or in deep soil, where it's often not coming in contact with some of these like man-made substances and chemicals. So that could be a cause of the variation. And another finding or thought is that alpha photobacteria has a big impact in converting N2 to NH3, which is ammonia. And for humans, we get our nitrogen intake from breathing through the air, but for many organisms, it comes from ammonia in the soil. So when we're destroying the ability for many bacteria, just like alpha protobacteria, to produce ammonia, that's gonna serve as a well-performing pesticide because it's causing all other organisms to not have enough nitrogen to function.
So for our findings for fipronil, we found that the Varroacomicrobia class reacts to fipronil in a way that increases their population size. With ASV8 and ASV17, they were both in the same order, family, and genus. I'm not going to pronounce all that. And they both increased in concentration from day 7 to 35. ASV14 is also Varroacomicrobia, but is a different in order, class, and order, family, and genus. And I noticed that it did also increase, but actually increased more significantly than with the other ASVs. So it does appear that there is a general increase in population with the Varroacomicrobia. And when fipronil is introduced, it just depends on it just depends on the species on how much it will increase. And this is probably because the fipronil is a molecule with that's a sulfoxide, and Varroacomicrobias who consume sulfoxides. So that's likely an energy source that's being introduced, and thus explains why the population is increasing in concentration.
So our findings for urea were actually pretty interesting. Something important to note is in both of these graphs, so we have ASV 72 and ASV 31, and both of those are known as rhizobiales, which means that they're nitrogen-fixing bacteria. And that means that they take in molecular nitrogen, which is N2, and they change it into ammonium. And that can decrease the pH and make the soil very acidic, which can be harmful to the microbial life. And we see that here because in both ASV 72 and in ASV 31, it's kind of hard to see, but right here we have urea, and you can see for the blue, that is actually the highest concentration of urea at 10x, and you can see at 10x here and at the 10x concentration here for urea, it's the lowest concentrations of the ASVs of our bacteria, which most likely means that too high of a concentration of urea can be harmful to the microbial life because too much nitrogen is being fixed and it's making the microbial life like too acidic. And can also be harmful because when it's too acidic, the proteins and the microbial life can denature, which means that they can change their shape and eventually lose their function. And this is also consistent in these graphs up here because so this was concentration, and up here we have days. And you can see for urea right here and right here, the concentrations of the ASVs did not change over time. And in both of these, since days was like our variable, concentration was controlled on both at 1x. And this most likely supports that the concentration of 1x or 1 molar is most likely the most ideal concentration, since over time, the concentration of our ASVs did not decrease. And then also our major accomplishments. Really our major accomplishments are just an overview of our project because none of us had really had any exposure to a project like this before with extracting DNA and using our studio. So of course our first step, creating our soil samples, we all got to learn how to use micropipettes, which was pretty intricate but also really fun. And something else important about our project is that each step built upon the previous step, which means we had to be very intricate and careful because if one step was wrong, that would really mess up the rest of our project. And so after, of course, we took our samples, we were able to successfully extract our DNA from our soil samples and check that we successfully did so. And then, of course, we used our 16S RNA sequencing to determining the genetic variation of our chemicals, and this was really significant because this provided our amplicon sequencing variant data, which we were able to input then into our studio to construct models of our ASVs over different periods of time and at different chemical concentrations. And then, of course, the last and most rewarding step was being able to take our models that we were able to take from our studio and be able to connect them to what we knew previously about our chemicals to try to better understand the underlying microbial functions and significance. And then here we are using micropipettes up here, and then here's like a 10x concentration of urea. But yeah.
And this is for our future directions, similar to how we conducted research similar to how our mentors did research on the Na22 project. Our findings also reached conclusive results, which means that with this same experimentation, none of these chemicals are what necessarily needs to be used to do this sort of experiment. So we can look towards agriculture and pollution. For agriculture, because of how more heavily reliant we are on fertilizers, and within our growing population we need more food, and for energy resources we need biofuel. Our experiment can be used to answer how the presence of certain chemicals and pesticides affect the health of soil and therefore the quality of food and crops. And understanding these effects can lead us to find strategies to mitigate risk associated with chemicals found in agricultural systems. On the other hand, we have pollution, and because pesticides tend to be more far-reaching after their application due to runoff from irrigation systems and bodies of water, our analysis can be used to investigate how specifically they are transported unintentionally after leaching, and we can determine if there are any traces of pesticides in the area. And specifically, we could even create models to predict how far-reaching they are exactly, and their effects on the soil and crops.
So the way we're gonna do this next part is one at a time. We're gonna stand at the podium, and if you guys have a question, that individual's gonna try to answer it. And don't be surprised if they may need help from their team or whatever, because all questions can't be foreseen. So we'll just kind of do that in order, and we'll see how that goes. Before we do go on, I did forget to introduce Liette, who is on a college visit for admitted students. So I do want to read Liette's introduction because she's also special to our group. Liette Zacharias is a senior at Lincoln East. She's planning on majoring in neuroscience, and she has been accepted to Brown University. Her ultimate career goal is to go into the medical field and become a physician. She is an avid participant in the Future Healthcare Professionals Science Club and Bring Change to Mind. She has also played tennis for the past 4 years at Lincoln East. Last year, she was awarded the National African American Recognition Award. She plans this summer to work as a certified nursing assistant at a nursing home. She also heads up as the leader, she's the president of Science Club, but she's also the leader of our Amazon project that we've been working on for about a year. So she's involved in so many different things. I just didn't want to forget that. All right, so why don't we just go in order as we go through. Anybody who has a question, just go ahead and raise your hand and then read your question if you got one. If not, you're going to have to get my harder question.
Is there a particular reason that you chose soil samples for Fermi Lab? Like why that particular?
Well, our mentors told us that it's a very I guess neutral kind of soil. Like it has like a good amount of all of the different nutrients, and like in Illinois in general, we have very good soil for things like crops, which is one of the big applications of this. So we wanted to test our soil in a environment or from an environment where it would be applied a lot, and by using the same soil across all the samples, it reduces the it reduces a variable that we
So
needed
was
to
that
know.
soil like what type of area was it? Was it like not having been agriculture before, or like you know what I mean, would not already have chemicals in it?
Correct. Yeah. It wouldn't. I mean, it would have had the same amount of chemicals as other surrounding soil, that would normally be encountered.
Like was it a prairie or was it a grassland or like just?
Do
you guys didn't know?
It was a prayer.
It was a prairie. Okay, it was a prairie.
Okay.
Yeah.
That's, I was just curious, like what type of soil?
Oh, sorry.
By the way, it's very well documented soil. They've done a lot of tests on it, so they knew almost everything about it. And that was another reason why they chose that soil for us to use in this, because of how well documented it is.
All right, so you were talking about the archaeobacteria. Was there a decision why you use archaeobacteria and not U bacteria?
She can be joined if another person wants to.
I feel like
I mean, the bacteria was more of like what we found using the RStudio. So we weren't initially trying to implement a certain type of bacteria into it. It was more like what the program produced based on the table that we were using, and then we used that to figure out what specific archaea may have been involved within this one.
And if I'm not correct, there was also there was U bacteria in there too. There was hundreds of millions, and we selected some to look at.
Okay, because I know archaeobacteria are extremophiles, and U bacteria can like general bacteria.
Good. Next.
I'm glad you're coming up, because I have an RStudio question. So, when I was in college, this was the big thing. And the procedure is very similar to what it was 20 years ago. But the one thing I'm not familiar with is this RStudio. When we were in college, the big thing was always Excel. What's the big advantage? Is it easier to import data? What's the advantage over other database programs? Why RStudio?
I mean, to be honest, it's just our mentor kind of said that this was the best I guess, application to use for what we were doing. I don't know if there was.
So R has been used so just like Python or like Python C, like R is a programming language and RStudio is like, they call it an IDE. So it's a tool that helps you with programming in R, and R has like a lot of libraries for working with soil. So it's a lot easier to use already existing and well-tested libraries that does a lot of the work for us. I mean, it would be thousands of lines of code if we had to do it all from hand, so.
By the way, to add on to that just a little bit, so Justin, he was sitting at the table. We had several days where we went to Argonne, and some were experimental days, some were data analysis days, and Justin's a PhD in bioinformatics, and there was some reason why our program was not giving us what we wanted. And in 30 seconds, I don't remember which one of you did it, but they saw the mistake in the code. To me, it could have been Mandarin. I couldn't see it at all. And they're like, oh yeah, you just forgot to put in this little symbol or whatever it was, and then it worked all of a sudden, which I could tell on his face he was immediately impressed by our team. So I just want to say that's proud of him. There's another hand sign.
How many species did you end up identifying?
We kind of chose there were thousands of them, so we ended up choosing the first 26 that had the most difference, that were different strains, because we checked out the DNA, checked it on blast to see if they were different, and I think we chose the first 26ish because the rest of them seemed to fall under the same phylum or family or order.
And the reason I asked was you said on the, sorry, I have no pair of glasses on, but was the Verruco microbacteria that you said the population actually went up?
Wait, can you say that one more time?
Sulfate loving one, the perma.
What was Sophia's slide?
Yeah, the protein.
Yeah.
So, and you said that that population went up, and your reasoning was that it was an increase in the food source over time.
Yeah.
And I was just wondering, from the different species that you had, obviously this is a complex explanation, but if you did any reading on possible competition from the other microbes that were found in the soil that might have, justified, perhaps those populations went down while this population went up kind of scenario.
Yeah, there's a good chance that has happened. I know a lot of our research we were looking at, I forgot which one of you, but with the chemical affects the pH, and then that affects the population. However, we were for this project we were looking more at individual bacterias to see what the chemicals effect was on that, and that could be something for further conclusions on top of our conclusions and further research.
Did I answer your question now?
Yeah.
Okay. That's it.
Someone mentioned a control. What was that control and where did that come from?
It was just 10
water.
samples, it was just
Where did that soil?
I think it was the same soil, yeah. It should have had no effects because there were no chemicals or.
Just treated with distilled water instead of treated with the exposure chemicals.
I have a few. You don't need to answer more than one. I'm sorry. So the final goal slide was for us to see how this could affect, well, how the soil microbacteria was affected by these chemicals, to see if those microbial differences was something that was negatively affected. I get, you go back to the original there should be more of a direct testing of whether or not there's a chemical presence of whatever, whether it's urea or that type of thing. Isn't there more, seems like a somewhat indirect way to test for, it's fascinating, and I think there are practical applications for it. It seems like a weird way to test for whether or not there was a pesticide.
So my question, I can see the application of this being is you're applying a pesticide as a farmer, how much is too much, or what combination of chemicals might have a negative effect on your microbial community? Just in
Yeah.
my mind, because you're looking at soil health, right? And
100%, right.
So maybe applying it to know how many different levels is kind of where I saw your whole presentation from my environmental focus, but
Yeah.
I could see that application.
I mean, actually like, doing this and knowing the link and all that, unbelievably awesome stuff, like crazy good stuff. One of the other questions was, so you have hundreds of millions of bacteria in the soil, right? Like there are, so you put them through this process of selecting for 26-ish,
Yeah.
right? It looks like we're trying to, what's that?
Yeah.
So you're kind of looking for this like, it's almost like a partial fingerprint, right? Because you're taking, you have this entire chain of bacteria, you're sequencing just a section of it. It's like a little bit of a fingerprint that then identifies that type of bacteria that you have, that you want to look for. And then it sounds like we selected the 26 that are, you know, the most affected, or gave us the best link to the data, but weren't there, you know, probably a few that were, you know, not affected, right? Or are there enough that we should show that, you know, this one doesn't seem to get affected? I'm not sure if there's.
So what's the question?
Well, I'm just saying, I guess how do you choose the 20? I guess somewhere in the presentation, we chose the 26 that were the most affected. And I guess the question is, how do we know that those bacteria that we chose are the ones that have some type of effect in the soil that would be either positive or negative?
Yeah, I mean, we kind of, okay, there's like a few parts of this question, but like definitely, like we'd look at the bar graphs first, and then we'd look back at the bacteria, and we would try to make connections from that, so it wasn't necessarily like, let's look exactly at these kinds of bacteria and look at the data. It was more so, let's just start looking through these graphs and see if we can find like patterns, and then look back and try to connect them to other graphs with similar like bacteria classes and like phylum and whatnot, to try to find the connections. And then also going off your question with like, how do we know which were like the most significant to look at? Well, there's definitely, I think as Davey was talking about earlier with BLAST, where we would match them up basically like nitrogen base by nitrogen base on this other platform to try to make sure like the ASVs we're looking at actually were like significantly different, and that it wasn't just like patterns due to like error and whatnot. But there's also a part of it, I know when I was looking at like ASVs like 31 and 72 is one of my lucky numbers, so I looked up like ASV 72 and there happened to be like a good pattern. So, but I mean, we definitely need to find more of like the direct connections.
I love your analogy of fingerprint, because you might be able to identify a fingerprint with just 4 or 5 points of reference, but if you don't, you can go to others. And so in the paper that they studied, the N22 paper, which was originally funded by Department of Defense, so that they might be able to go to a facility, see, take a soil sample, and using references from soil being exposed to any number of contaminants. It doesn't have to be, we chose these because they're not dangerous to students. But there's so many things that could change all the microbacteria, and once you have all those, now you have the fingerprint for that exposure. And so you could go to any facility, reference that fingerprint, and see, was that facility exposed to that substance? And that's the application ultimately that inspired this type of research, but it's going much further than that. Like they pointed out in their future directions that we could say, all right, we tested this riverbed and now we found the traces of this substance has made it from the mountains of a mine all the way to, wherever we tested it. So basically, you can go anywhere and see what exposures have happened to that particular area.
Yep.
I guess to build up off of this question, are we skewing data in a sense, not just picking the 26 that are most affected? But like, how do those 26 relate to whatever hundreds of thousands of different types of bacteria were in the soil? Like, do they represent the majority of bacteria that's in that soil? Or you would just say, I want to pick the ones that look the most statistically significant? Because then I'm just skewing data one way or the other. And if I incorporate the other bacteria, is it going to look more normalized?
Well, the point of the study is really to identify what has the biggest variance. It's because, as he was saying, with that variance, we can see what it has been exposed to. So there are thousands and thousands that have barely, if at all, been affected. But by looking at the 26 that have been affected the most, we can identify what has been exposed to that.
And if a database is created, they wouldn't stop at 26. This is a demonstration of the concept, for these young people to inspire them to move on in a direction. But in terms of the governance database, they would probably apply it to probably 10,000 species. But in this case, because we only had so much time to devote towards this, we had to abbreviate it.
More from a comment, you guys just indirectly prove what scientists automatically do. They always look for what's going to happen to the outlying group, the extremophiles, because they're going to show the biggest change. So by selecting what you did, you just kind of proves that we're going to look at the group that's going to have the biggest change and see what happens to them. So if you would have dove into the middle, you might not have seen anything, or you might have seen huge changes. So you indirectly just suggested why we look at specialists when we look at environmental problems, because they're the first we act.
This is more about like the last slide. So I know just from having my sister who works in like food science, that a big problem with agriculture is the depletion of nitrogen and things, and how to turn that over, et cetera. So what did you notice about the effects of the pesticides in general on like, the nitrogen in the soil that could be used for agricultural purposes?
So we know that urea was our nitrogen sample. So we could see that any bacteria that would feed off nitrogen would increase. But it'd be hard to find actual correlation between those other chemicals, specifically with nitrogen, just because there's no real nitrogen like tracker. What we could do is we could see, hey, this bacteria reacted heavily to urea. Let's see how atrazine, fipronil, gamma affected that same bacteria, and then we can make some conclusions from there.
Also, once one species proliferates, other ones will diminish. And it may not be a relationship to the substance we put in, but just because of opportunity or because another bacteria is not consuming this nutrient, another one will now capitalize on that opportunity. So just the act of one thing killing another might cause another one to grow, but that's all part of how they're all interrelated to each other.
Mr. Murphy?
I need to put you on the spot. Who's your favorite science teacher? No, I just want to say you guys did a great job. You make us proud, so keep it up.
Thank you very much. Any other questions, Mr. Billy?
There was one of the pesticides that said that they didn't have known effects or known effects in humans or whatever. How does the FDA let that slide, or is that are you able to use that as a pesticide and not know what the overall effects are? Or is it just not? Or is it denied? I think that was the fipronil or something.
I think both of us said something like that.
We know that benzene is going to cause
Cancer.
So that needs a little more clarification. So that's like what's in Roundup and a lot of the other things they found to be really
Yep.
Yeah, let's keep in mind, this is not, our study was not a study of toxicity for humans. It was only to see the relationship of stressors on soil to changing the bacteria DNA profile, so that you could apply this to any stressor put on a soil. But even if it was something that would be considered a benefit, let alone maybe something that would kill bacteria or kill pests.
So I don't know if you looked at this or just something like, I was wondering, you said in your data that gamma ended up increasing the soil decomposition.
Yes.
But how does it do that if it's acetylcholinesterase inhibitor?
Okay, so I believe that I did not look at this, but it was something with the property of acetylcholine interacting with our soil that allowed certain bacteria to grow in the soil, I believe, and that's why they created the decomposition of, or increased rate of decomposition of the soil.
By the decomposition of soil, of course, there's organic components, but there's also inorganic components. So, anything that's an organic component can turn into a variety of different substances. So, each one of those, is somewhat unpredictable, for us in this case.
In terms of the procedure, and did you pick a sample here, you were gonna focus on this particular one, and then carry that whole procedure out yourself, find the molarity? Did you guys team up? Was this individual? Did the scientists make the solutions? Like, what exactly, when you talk about that procedure, what was your part of that?
So we came, did we come 3 separate days, I think?
Yeah.
And we came in different groups, and we just we divvied up all, because we had to do different molarities, different, and all the different chemicals. So we just divvied up the different processes across the 3 days, and everyone had a direct hand in creating the samples, and the mentors just guided us through it, but we did do all the work of the micropipetting and the
Did they double
centrifuging.
check your work?
I mean, they were watching it the whole time, and I guess a benefit of the 16S sequencer is it showed us I guess there was a percentage of how well we did. I don't remember the exact what the name of the measurement was, but it showed us the percentage of twigs and stuff that was still in the sample, and we had very good numbers, which would allow us not have much anything outside impacting the DNA we collected.
Don't forget, there was a scan that we performed after we did the extractions to make sure that the extraction was rich in DNA before we sent it off to the sequencer. The sequencer was the expensive portion, and so they didn't want us to send samples that were going to waste their time. So yes, we did have a scanning process to make sure that our extractions, we didn't mix up in the kit. A step, there was some of the kits had 20-some steps to them. So in that process, they want to make sure that our extractions were effective.
This is a general question, and I don't know if you can answer it, but just your overall experience, what was your favorite part of the experience? And did it solidify or even shift kind of your focus? Because many of you are looking towards a degree in science or a career in science. Did this change what you were thinking initially, or kind of solidify where you're thinking for the future? What was your favorite part?
For me personally, I just really liked working in the lab. Just being in the lab was kind of cool, and I already had an interest towards pharmaceutical drugs and how important it is in our society because it is medicine. And I think it really did solidify what I wanted to do, but more so originally I just wanted to be a normal physician, and then once I realized how cool being in the lab was, that's when I decided oh hey, maybe pharmacy is kind of cool to get into. I don't know about everyone else's experience.
Did it change for anybody? Would anybody like to speak up on that?
I mean, I know originally for me, I was just planning on getting 1 degree in engineering and then going off of that. But I decided then I'd add a bit of a chemistry degree because I really enjoyed the lab work. And even though the programming was completely new to me, it it was a challenge on a school computer, but I but I was able to like go through it. It just was very rewarding for me, and it's something that I would definitely do this all again if I could.
I mean, I guess I can kind of like speak on this too, kind of going off what Sophia said. It was like very interdisciplinary, and I like originally was thinking going into more like hands-on like biology, but being able like doing the whole data analysis part of it with like our studio was like really interesting to me as well. Like being able to make the models and like look at all the different patterns. I thought it was like really exciting to be able to analyze our data in that like sense, and so I was kind of thinking of maybe more minoring in something with like data analytics or something like that, just because it was like really exciting to see like how our whole project came together with that process.
Our two scientists complement each other. One was an expert in lab techniques, and she was a big part of everything COVID for Argonne. So testing techniques, disinfecting techniques, she tested all those different things. But then her partner, Justin, he was a computer wizard, and both of them very personal people that made this super interesting in every aspect. And I was really pleased to see how many people said they were interested in bioinformatics because that's what Justin's PhD was in. And I thought, he had a big influence in this, maybe driving some of them towards that. Some of them probably had that already because they wanted to be in computers. But to see it in action, I appreciated it myself.
Okay, so you guys were tracking the populations of the microbials based off the chemical, correct?
Yeah.
Okay. My question is kind of about your mentors. For your mentors, like how much for them looking for radiation, how much like carried over in terms of your procedure? So I guess what I mean, like, did your mentors do the same thing, like looking in soil for microbial populations based on how it reacted to radiation? I don't know if you would know, or were they looking for radiation signatures within the soil?
So they used, didn't they use like carbon or like I don't remember what the KT is. What was it?
They didn't actually like look for like the radiation in the soil. They looked for presence of or the effects of the chemicals used in like creating the radioactive material. So it's like the chemicals themselves that they were like how we had urea or gamma, theirs were not actually radioactive. They were just other chemicals. I don't remember.
I think it was like carbon related more so, and they used, but the result, the idea of what they were analyzing was the same. Though they wanted to see how variations in the microbacterial communities in the soil changed based on what the concentrations they used and the days. So the data analysis part was the same for the most part, but what they used was, I think it was more carbon related, whereas we used pesticides and insecticides.
When people are processing high energy materials like uranium and so forth, the Purex chemicals are common, and they're volatile, and they make it out the door of facilities. And so they were looking for in the soil evidence of the use of the Purex chemicals for the extraction of weapons grade plutonium and uranium. And so they found that correlation that they didn't have to go in the buildings because a lot of countries don't let us in their buildings, but they'll let us walk around their buildings. You don't even have to, they could have moved everything by that time, but the soil samples will still show the presence of those things, and that was the idea behind. Now, nuclear exposed or radiation exposed soil will have a different set of factors than the Purex chemicals would have on the soil. Anybody else?
I got one. So for the process, more of a process. So based on your research, what question would you like the next research?
I mean, I've really found it interesting how each of the different microbial communities and different bacteria, how they reacted, and I feel like it'd be more interesting to explore more into that, like see if, say, like if the nitrogen's really affecting it, if we tried other fertilizers that were more nitrogenous and less like us looking in general, like if we could find more concrete evidence to what we have figured out. And I just think that'd just be really interesting to have everything confirmed and also look a bit further and see, maybe we didn't see it with with the fertilizers we use, but maybe there's another bacteria that is significantly affected by some other chemical we did not test on.
So this is a question I you can answer, but what would you do differently now that you know what you've gone through in 6 months?
I feel like it was, if we were able to do it again, it would be interesting to kind of look at how like maybe the different chemicals like reacted with each other, because we looked at it more like one at a time of like urea and atrazine and fipronil and whatnot. But I think it'd be interesting to look at more. I think someone brought this up earlier about like competition between chemicals and like the different bacteria, and to look at it more like interdisciplinary instead of just like taking one at a time. I think would give us really interesting results.
All right, looks like they've exhausted your questions. Super pleased that you were able to give such well thought out questions to them. They're going to get those kinds of questions in a couple days, and now they've had some practice hearing them and thinking about that. Thank you so much for coming.